Hi friends, welcome back to my channel. So today let's start with the class, continuation of the eukaryotes. Lysosomes and peroxisomes are containers of degradative enzymes. So from here we will be seeing in this class. Lysosomes, which were discovered in 1949 by Christiane D. Dewey, are organelles bounded by a single membrane that are of variable size and morphology, although most have diameters in the range 0.1 to 0.8 micrometers. So in 1949, Christiane D. Dewey, he discovered lysosomes. So lysosomes are the organelles which are bounded by a single membrane that are a variable size and morphology. So most of the lysosomes having a diameter in the range 0.1 to 0.8 micrometers. Lysosomes, which are essentially membranous bags containing a large variety of hydrolytic enzymes, function to digest materials ingested by endocytosis and to recycle cellular components. So here lysosomes are essentially membranous bags containing a large variety of hydrolytic enzymes. So these lysosomes contains a hydrolytic enzymes present inside the membranous bag. So these lysosomes function to digest materials ingested by endocytosis and to recycle cellular components. Cytological investigations have revealed that lysosomes form by budding from the Golgi apparatus. So cytological investigations have revealed that these lysosomes are formed by a budding from the Golgi apparatus. Peroxisomes, also known as microbodies, are membrane-enclosed organelles typically 0.5 micrometers in diameter that contain oxidative enzymes. So these peroxisomes are also known as microbodies. So these peroxisomes are membrane-enclosed organelles. So they typically range from 0.5 micrometers in diameter. So they contain oxidative enzymes. So these peroxisomes contains oxidative enzymes. They are so named because some peroxisomal reactions generate hydrogen peroxide. So these peroxisomes are so named because some peroxisomal reactions generate hydrogen peroxide. H2O2 is an hydrogen peroxide. A reactive substance that is either utilized in the enzymatic oxidation of other substance or degraded through a disproportionation reaction catalyzed by the enzyme catalase. So here a reactive substance which is either utilized in the enzymatic oxidation of a substance or degrade through a disproportionate reaction catalyzed by the enzyme catalase. So by utilizing the enzyme catalase, the reaction is produced. 2H2O means 2 hydrogen peroxide gives rise to 2H2O plus oxygen. So 2 molecules of water and 1 molecule of oxygen is released from 2 molecules of hydrogen peroxide. It is thought that peroxisome functions to protect sensitive cell components from oxidative attack by H2O2. So through peroxisomes, which is functioning to protect sensitive cells, they are having components from oxidative attack by hydrogen peroxide. Certain plants contain a specialized type of peroxisome. So there are certain plants which are specialized type of a peroxisome. So they contain a specialized type of a peroxisome. The glyoxisome, so named because it is the site of a series of reactions that are collectively termed the glyoxylate pathway. So glyoxysomes are so named because it is the site of a series of reactions collectively termed as a glyoxylate pathway. Site of a series of reactions that are collectively termed the glyoxylate pathway. So this glyoxylate pathway takes place in the peroxisome. So next, E. The cytoskeleton organizes the cytosol. Cytosols, far from being a homogeneous solution, is a highly organized gel that can vary significantly in its composition throughout the cell. So the cytosol, which is being a homogeneous solution, is a highly organized gel. So cytosol contains a highly organized gel very significantly in its composition throughout the cell. Okay. Much of its internal variability arises from the action of the cytoskeleton, an extensive array of filaments that gives the cell its shape and the ability to move and is responsible for the arrangement and internal motions of its organelles. So there are an internal variability arising from the action of a cytoskeleton. Okay. To maintain the cytoskeleton, an extensive array of filaments which gives the cell shape and ability to move and is responsible for the arrangement and internal motions of its organelles. Okay. So this cytosol is helping in the movement, okay, arrangement, shape and internal motions of the organelles. So the cytosol is in the form of a fluid form. Okay. The most conspicuous cytoskeletal components, the microtubules, are approximate 250 angstrom diameter tube that are composed of the protein tubulin. So the most conspicuous cytoskeleton components are microtubules. Okay. So these microtubules are approximate 250 angstrom diameter tubes. Okay. So they are composed of a protein tubulin. Okay. So the microtubules are formed with the composed of a protein called a tubulin. Okay. They form the supporting framework. They form the supportive framework that guides the movement of organelle within a cell. So this microtubules, so these tubulins form the supportive framework that guides the movement of the organelle within a cell. Okay. For example, the mitotic spindle is an assembly of microtubules and associated proteins that participate in the separation of the replicated chromosomes during cell division. So these tubulins are working as a supportive framework which is guiding the movement of the organelles within a cell. Okay. Example, mitotic spindle. If we see the mitotic spindle, it is an assembly of a microtubule and associated with the proteins that participate in the separation of the replicated chromosomes during cell division. Right? So microtubules are also major constituents of cilia, the hair-like appendages expanding from many cells, whose whip-like motion move the surrounding fluid past the cell or propel single cells through solutions. So these microtubules are major constituents of the cilia. So they are like hair-like appendages. Okay, so they are extending from many cells. So the cilia are looking like a hair-like appendages. Okay, whose whip-like motions move the surrounding fluid past the cell or propel single cells through the solution. So this is about the cilia. Very long cilia such as a sperm tail are termed flagella. So if we see the flagella, they are having a very long cilia such as a sperm tail are termed as a flagella. Okay. Prokaryotic flagella, which are composed of the protein flagellum, are quite different from and unrelated to those of eukaryotes. So the prokaryotic flagella is composed of a protein called a flagellin. Okay. So these flagellins are quite different from and unrelated to those of the eukaryotes. Okay. The microfilaments are approximate 90 angstrom diameter fibers that consists of a protein actin. So if you see the microfilaments, they are having approximate 90 angstrom diameter fibers. Okay. So these fibers that contains a protein called an actin. Right. Microfilaments, as do microtubules, have a mechanical supportive function. Furthermore, through their interactions with the protein myosin. So microfilaments are do like a microtubules, okay, have a mechanical support function. So these microfilament are involving in the mechanical supportive functions, okay. Furthermore, this microfilaments are interacting with the protein called a myosin, okay. Microfilaments form contractile assemblies that are responsible for many types of intracellular movement such as cytoplasmic streaming and the formation of cellular protuberances or invaginations. So if we see the microfilaments, they are forming a contractile assemblies that are responsible for many types of intracellular movements, okay, such as the cytoplasmic streaming and the formation of the cellular protuberances. Okay. More conspicuously, however, actin and myosin are the major protein components of the muscle. So if we see the actin and myosin, they are the major protein components of a muscle. Okay. The third major cytoskeleton component, the intermediate filaments, are protein fibers that are 100 to 150 angstrom in diameter. So if we see the third major cytoskeletal component, it is an intermediate filament. Okay. So this is the protein fiber that are having a 100 to 150 angstrom diameter. Okay. Their prominence in part of the cell that are subject to mechanical stress suggests that they have a load-bearing function. So this intermediate filaments is having a prominence part of the cell. Okay. So they are subjecting to the mechanical stress, suggesting that they have a load-bearing function. Okay. So they are having a load-bearing function. For example, skin in higher animals contain an extensive network of intermediate filaments made of the protein keratin. So if we see the skin of an higher animals, they contain an extensive network of the intermediate filaments, okay, which is made up of a protein known as a keratin. Okay, this keratin, which is largely responsible for the toughness of this protective outer covering. So there is an outer layer covering to this keratin. Okay, so this keratin is having a very toughness of the protective outer covering. Okay, in this contrast to the case with the microtubules and microfilaments, the protein forming intermediate filaments vary greatly in size and composition, both among the different cell types within a given organisms and among the corresponding cell types in different organisms. So in contrast to this microtubules and microfilaments, the protein forming an intermediate filament, they vary in size and composition. Okay, both the microtubules and microfilaments among the different cell types with a given organism and among the corresponding cell types is different types in different organisms. Okay friends, thank you. Thanks for watching. If you like my video, please kindly like, share and subscribe it with your friends so that it would be helpful for them also. So in the next class, we will be studying about the plant cells are enclosed by a rigid cell wall. Okay, from here we will be continuing in the next class. Okay, thank you. Thanks for watching. We will meet in the next class. Bye bye.